always post the recording to YouTube, so I'll send you guys the link when I make the video so you can watch it ahead of time. I understand that, this morning we had a bunch of thunderstorms. I know some people lost power, and I understand that with everything going on, you might have had to pick up a job or, work extra shifts. And so for attendance purposes, you do not have to be present in WebEx for this last unit. I want you to learn the material, I want you to get the material, but it is not going to change your attendance points whether you come to every WebEx or not. And when I go through Canvas, I'll show you what to do if you miss, or how I'm going to grade attendance. So if at any point you have questions, please put them in the chat. I'm going to first start by going to Canvas here. Make sure I can see the chat. Okay, so this is your Canvas view. We have that updated syllabus. If you go all the way down here to material covered for exam 4, I have first the PowerPoint that I'm going to be using. These are all the slides for the whole unit. So if at any point you want to go back and look at the slides again or follow along, however you like to learn, those are going to be right there. I have also put the assignments for Unit 4. And there's half of them are due this week, half are going to be due next week approximately. And I'll go through those briefly with the assignments, but they're all here. And then I put some helpful videos that I think can give you some more information if there are topics in this unit that you find confusing, some extra resources for you. I also made a heading for the lab. So there's some background slides for the lab material, what we're going to be covering in lab. And for each lab, there are 3 lab activities we're going to do, and they're short. Basically, I want you to read the worksheet. I give you some data tables. I want you to do some calculations, and then each worksheet will tell you what you have to submit each week, and each one is due each Thursday. Okay. So if we're going to the assignments page, I just want to show you where everything is so you can find it. Upcoming assignments, the first thing you have to do, and this is due on April 16th at midnight, is a pre-lab quiz. So if you click on the quiz, it gives you the full handout. So I took all 3 weeks and I pushed them into one document. So I want you to first read through what you're going to be doing for the 3 lab activities, and then take a short quiz on that, just like a normal pre-lab quiz. Okay, that's the first thing that's due. Then you have your first lab activity due on April 16th at midnight as well. So if you click on this link, it gives you the first handout. And on Thursday, I'll post a short video going through some background slides for you. But we won't meet in the WebEx room for lab. So for lab, there's no meeting time. You just have to complete each part of the worksheet. So for this first one, it goes through. Well, either way, you can look at the first one and it breaks down the data you need to calculate and then what you need to submit to Canvas. And I'll post a video by Thursday explaining those things, the concepts, what you need to do. Okay, so look for that video to help you with this first lab assignment. Now, you can do all the lab assignments at once. They're not that long. There are specific segments due each week, but they're open for submission if you want to get them all done ahead of time. More power to you. Okay. The other things due this week, you have a short homework about the kidney and about how kidneys are different in different animals. So I want you to download this document and then submit it here using your textbook to answer how are kidneys different? How do different animals meet their needs with this structure and how do they compare? So a comparative anatomy type of homework. There's also a short quiz that you should use your book to help you with about ionic and osmotic regulation. And that's going to be the first thing we'll talk about today. I also want you to submit a drawing. So you're going to draw a nephron, label different parts, and this will make more sense after Wednesday's lecture in particular, and submit that. Okay. And then that is the first week. Second week, you've got another lab assignment, another drawing. What pages will help us with the renal homework? Okay, so on the homework itself, it tells you it's the end of chapter 7. So at the end of chapter, or not chapter 7, I just lied to you. The end of chapter 10. The homework is based on the end of chapter 10. And there's also some slides in the PowerPoint about it too. So if you go to the end of the PowerPoint, you'll see that. Basically, it's about how different animals, what the kidney looks like in different animals. So it's the end of chapter 10. It's a good question. Okay. These are all the assignments that are put here. The last thing I wanted to just bring up was the discussion question. So I don't know how Dr. Pogue was doing attendance or what he was counting for attendance. For this unit, I'm not going to take attendance in WebEx, and all I want is for everyone by the end of the unit to participate in this discussion. So to answer these 8 questions, they're longer. So you can get them done whenever. You can start them now. You could put them off to the end. Either way, your attendance point are completing these 8 questions. You don't have to get them correct either. So I won't take off attendance points if you make a mistake with these questions or you miss a component. So the attendance grade, again, for this unit will be whether you, at some point before April 27th, answer these 8 questions. Okay. I'm going to switch gears here. Are there any questions about how I'm grading things, what you have going on for this unit? Put them in the chat. Okay, while you think about that, I'm going to pull up the PowerPoint. And my goal for each lecture is to not talk for the whole 50 minutes. So my goal is to share, spend about 30 minutes, 20 to 30 minutes on a lecture, and then give you the rest of the time to do all these other activities that I think are more beneficial for learning. So I'm gonna start there. Again, this is the 4th unit. We're looking at renal and urinary physiology. So these are the outline of the topics. Dylan, to your question about page numbers, right here it says comparative anatomy is broken into 2 different sections. So there is some comparative anatomy at the beginning of chapter 10, pages 472 and 474 to 478, and then 45 to 49. And then there's some at the end of the chapter, 514 to 518. So you'll need to look at the very beginning and the very end of chapter 10 for the comparative anatomy component. The first unit, we're going to talk about ionic and osmotic regulation. And we're going to identify the goals of ion and water systems, and then we'll distinguish between conformers and regulators and talk about a little bit about what happens in fish, reptiles, and birds. So when we're thinking about water balance systems, we want to think about the goals of these systems. And there are a few different goals that animals need to meet. So they first need to have osmotic regulation, which controls the driving force of water across membranes. Remember, it's easier to move salts than water. It's easier to move salts than water because water is going to use aquaporins. And remember, with aquaporins, water is going to just follow its concentration gradient. And so it's easier to use a pump, for example, and move salt where you need it to move, and then have the water follow. So that's generally the strategy animals will use, is to think about osmotic regulation as moving salts over water. So the other goal that they need to have is ionic regulation. So again, controlling the salts. Someone's microphone is not muted. Oh, okay. Great. Any questions at this point? No, okay. So 3 goals. We have osmotic regulation, we have ionic regulation, and then the other goal is the nitrogen excretion component. It's to excrete ammonia. Keep in mind, ammonia is a toxic byproduct of protein synthesis. So you want to think to yourself, as you look at different systems, how are they meeting these 3 goals? How are they meeting the goals of osmotic regulation, ionic regulation, and nitrogen excretion? So with this in mind, you want to think back to tonicity and what happens to cells at different tonicity. So hypertonic solution, right? That means that there's a lot of salt outside the cell. And again, the water is going to try to move to decrease that saltiness. So it's going to leave the cell in a hypertonic solution. It's going to get very shriveled. Isotonic, meaning the concentrations are the same. The net movement of water inside is going to be equal to the net movement outside. And then in a hypotonic, this is going to be the reverse of that hypertonic solution. So outside, not a lot of salt. Inside is considered very salty, and the water is going to flow in. So you want to keep that in mind when you're thinking about osmotic regulation. You also want to think about the environment that the animal is living in, especially when we're talking about different kinds of fish. So marine animals have an environment that is high salt. They have lots of salt in the water, more so than in their body. And what their goal is, or what the driving force they're going to need, is they're going to need to obtain water against a gradient. They're going to need to pull water out of a very salty solution. Whereas freshwater animals are in the opposite solution. They need to, instead of pulling water against its gradient, they need to somehow get salts out of a hypotonic solution. And so to do this, we're going to look at how there are sites of ion and water exchanges, both on outer coverings, the respiratory, and the digestive tracts, and what's happening at these sites of exchange. So there are two general strategies, conformers and regulators. Conformers let their internal conditions vary with the external conditions. So they just conform to their environment. These are the animals that give into peer pressure. They're going to just say, whatever the concentration is outside of me, that is what I am going to be. Whereas regulators maintain a constant internal state despite a changing environment. So they're going to maintain their internal conditions no matter what kind of environment or surround they're in. And this can be both for osmo, for water, and for salts. So ionoconformers versus regulators. Again, ionoconformers, they have little control over their internal solute concentrations. These are usually some seawater animals like cnidarians. Other marine animals are going to take an ionoconformer type of approach. Whereas most advanced organisms regulate their internal conditions. They have control over their ion balance. They're regulators. Okay. This is showing this in a graph. In science, we love to graph things. So if you were to graph the external osmolarity, so the saltiness in the environment on the x, as a function of that internal osmolarity on the y here, it's showing you that osmoconformers, things like jellyfish, which are other primitive vertebrates and marine invertebrates, those are going to be osmoconformers. They're going to let their water concentrations, their external osmolarity and their internal osmolarity are going to match. So you can see that in the yellow line. I guess it's yellow, maybe yellow-orange. As they are exposed to higher and higher osmolarity environment, their internal osmolarity also rises. Whereas osmoregulators, things like mammals, for example, shown in purple, they maintain that steady osmolarity on the inside despite a changing environment. Okay, conformers versus regulators. Okay, now let's talk a little bit more about fish. Before I move on, are there any questions? Again, if at any point you have a question, put it in the chat feature and I'll stop and answer it. Otherwise, I'm going to just keep going. So fish gills transport ions into and out of the water solution. So we want to look about, when we think about fish, you want to keep in mind that we have differences between marine fish and freshwater fish. Okay, but in general, the gills are going to be where the site of exchange for ions. And they have these mucus secreting cells on the surface of their gills that keep it a lubricated surface. And then they have two different cells in their gills, both chloride cells and pavement cells. And chloride cells, when you look at them, they're larger, they have a lot more mitochondria. It's shown here in the electron micrograph picture. All those little dark regions, those electron dense regions, those are going to be your mitochondria. So you can see very high energy demand filled with mitochondria, whereas the pavement cells are smaller and flatter cell. And so the gills of freshwater fish, again, they're freshwater, they're living in a hypotonic solution. Their goal is to take up salts. They want to take up ions. And so they're going to use both the chloride cells and the pavement cells. The pavement cells, you also see them referred to as PNA positive cells. These take up the sodium through their apical channels and then use energy to maintain that gradient in the cell. Whereas the chloride cells, we call them PNA negative cells, they are the ones that take up chloride with a bicarb exchanger. So HCO3 is going to be produced in the cell, and that's going to drive the gradient. Whenever you look at the diagram I'm about to show you for the cells, you want to keep in mind what is driving the movement of these salts. So what is providing the energy? Is it actual ATP? Is it the gradient of some other salt or solute? What is providing the energy source? Okay, so if we look at the chloride cell here, in marine fish, they're going to need to limit their water loss. So marine fish, again, they have a bunch of salt around them, and instead they're spending their energy to take in water. So they're going to use that sodium potassium chloride cotransporter to build a gradient, and then the chloride will be able to diffuse through the cell into the water. So this is what that is showing you here. On the top, we have the seawater side, and on the bottom, you see the blood side. And they're again using that channel to build the gradient, the cotransporter rather, to build that gradient. Okay. This is showing you both freshwater and marine side by side. So if we look at a PNA positive cell, PNA negative cell, you can see shown in blue here is the water, okay? And the inside here is that pink color. And so for the PNA negative cell, they're going to use sodium gradients to drive, or they use the bicarb they make as a gradient to drive in the movement of sodium, okay? Whereas in marine fish, their goal is going to be different. So for all of them, you want to keep in mind, what does the environment look like? What's setting up the gradient? What's driving that? And for this section in particular, I also want you to make sure that you read that first section of your textbook about conformers and regulators, okay? And so on Canvas, the quiz that I set up is going to be about the different strategies. So you want to make sure you read your book well for that section, you take notes, and then you go and answer those quiz questions. Switching from fish to reptiles and birds use salt glands. So salt glands are going to excrete a hyperosmotic solution of sodium and chloride. Hyperosmotic, very salty solution. And this allows them to derive water from seawater or even sometimes from food. So some birds can get water, especially desert birds, for example, can get water from their food and they don't need to actually take in a water source. And this solution can be 3 times as concentrated as the blood. So if they take in 30 mils of seawater, they can excrete 10 mils of concentrate and then retain 20 mils of pure water. And this is really helpful for birds like seagulls, for example, that are around a very salty water solution. And this is what the salt gland looks like. So shown on the top is the blood vessel, and the blood is going to flow. And they'll be able to use transporters to take in those sodium and chloride molecules and make this super salty solution in their salt gland that they can then excrete. Okay. This first one, switch gears here. Okay, so this first one is a little short on purpose because what I want you to do for the rest of the class time is to read your textbook, which I know is quite possibly the most boring thing I could tell you to do. But I mean it. I want you to read your textbook and take notes on this first section. What I gave you was just an overview. And in order to answer the quiz questions, you got to go into a little bit more in depth and get some more details on it, okay? So I want you to take the rest of the time, because this was only 20 minutes. So I want you to spend a half hour reading your textbook and then going through and answering that quiz. Now, the quiz is not due right now, it's due in a little bit, but I think that would probably be the best use of your time. With that, I'm going to end the recording for today, and I